Site-directed mutagenesis is useful to study molecular interactions such as RNA-RNA and RNA-protein interactions. This protocol describes how to perform site-directed mutagenesis with a 2- or 3-step PCR approach. The main advantages of the method are the diversity of different mutations that can be incorporated, as well as the relative ease and cost for doing so.
To begin, use the wild-type DNA template and primer pairs 2-1 and 2-3 specific for 2-step, or primer pairs 3-1 and 3-4 specific for 3-step PCR to obtain PCR product 1. To validate PCR products by agarose gel electrophoresis, make a 2% agarose gel by dissolving 2 grams of agarose in 100 milliliters of 1x-TAE buffer using a microwave oven. Add ethidium bromide at a final concentration of 0.5 microgram per milliliter. Then, cast the agarose gel. When solidified, place it in an electrophoresis unit. Load a DNA ladder of known size and the PCR samples mixed with DNA loading dye into wells. Run samples at 75 volts for 45 minutes and visualize bands on a UV transilluminator or with a gel imaging system. Next, purify the PCR product with the gel extraction kit according to the manufacturer's protocol and measure the concentration of the purified DNA with a spectrophotometer. Then, store the purified DNA at -20 degrees Celsius in TE buffer. To begin site-directed mutagenesis with a 2-step PCR, first use the wild-type DNA template and the primer pairs 2-1 and 2-2 for 5' end mutations, or primer pairs 2-2 and 2-3 for 3' end mutations to obtain PCR product 2. Validate the PCR product by gel electrophoresis, purify the PCR product, and measure its concentration as described before. Then, use the wild-type DNA template and the PCR product 2 as the primer together with primer 2-3 for 5' end mutation, or primer 2-1 for 3' end mutation, to obtain PCR product 3. Validate the PCR product by gel electrophoresis, purify the PCR product, and measure its concentration as described before. Then, store the purified DNA at -20 degrees Celsius in TE buffer. To begin site-directed mutagenesis with a 3-step PCR, first use the wild-type DNA template and primer pairs 3-1 and 3-2 to obtain PCR product 2. Use the wild-type DNA template and primer pairs 3-3 and 3-4 to obtain PCR product 3. Use the wild-type DNA template and primer pairs 3-3 and 3-4 to obtain PCR product 3. Validate the PCR product by gel electrophoresis, purify the PCR product, and measure its concentration as described before. Finally, use 2 to 5 nanograms of PCR products 2 and 3 as the template, along with primer pairs 3-1 and 3-4, to obtain PCR product 4. Validate the PCR product by gel electrophoresis, purify the PCR product, and measure its concentration as described before. Store the purified DNA at -20 degrees Celsius in TE buffer. In this study, 2-step and 3-step site-directed mutagenesis were used to investigate RNA interactions regarding post-transcriptional regulation of CSGD. The wild-type CSGD was PCR amplified to yield PCR product 1A, and the wild-type MCAS was PCR amplified to yield PCR product 1B. Using a 3-step PCR strategy, PCR products 2A and 3A were synthesized in the first 2 steps, and 4A in the 3rd step to introduce mutations in CSGD. Similarly, complementary site-directed mutations were introduced in MCAS to yield PCR products 2B, 3B in the first 2 steps, and 4B in the 3rd step. Western blot analysis showed that while expression of wild-type MCAS prevents translation of wild-type CSGD, mutations in either MCAS or CSGD alleviates the observed repression. However, mutations in both CSGD and MCAS prevent translation of CSGD. Using a two-step PCR strategy, PCR products 2C, 2D, 2E, 2F, and 2G were synthesized in the first step, and PCR products 3C, 3D, 3E, 3F, and 3G in the second step to introduce mutations to the entire CSGD DNA. EMSA results of the HFQ binding to in vitro transcribed CSGD wild-type and mutant RNAs, synthesized using PCR products 3C, 3D, 3E, 3F, and 3G, showed the increased KD values of the mutant alleles. This proved that HFQ could bind less efficiently to the mutants. Furthermore, HFQ has several binding sites on CSGD, which is shown by the 3 shifts observed for the wild-type RNA. However, only 2 binding sites are observed for different mutant RNAs. Thus, the site-directed mutational approach identifies primary and/or secondary structures of the CSGD mRNA that are important for complete binding of HFQ.
The method for site-directed mutagenesis described here relies on PCR. Good PCR practices are therefore crucial for the method and might require optimization to be successful. Site-directed mutagenesis is only powerful with downstream methods such as EMSA and Western blotting. Other methods, for instance, include various structural assays, enzyme activity assays, and post-translational modification studies.